What do you think of when you see a pineapple? Medicinal properties probably do not come to mind. However, current research has linked bromelain, an extract from this fruit, to potential treatments for cancer patients. Virtually non-toxic and lacking in any negative side effects, this naturally occurring compound has proven to have successful applications in epidermal carcinoma oncology, based on its anti-inflammatory and therefore anti-proliferative properties. In this way, bromelain is a worthy subject for further research in the medical field, as it has great potential to offer a significantly less harmful, more readily available, and more natural alternative treatment to cancer patients than current chemotherapeutic practices. Found in the stem and core of the fruit, bromelain is a cysteine protease that is used by the pineapple to derive nitrogen and phosphorus from organic material. Determination of the structure of this hydrolase enzyme is still within its theoretical stages, although the proposed 3D representation is recognized by PyML software under the theoretical PDB code 1W0Q. Although the medicinal applications of bromelain have been widely recognized, the specific mechanism through which it acts was not understood in great detail. In humans, bromelain has been used throughout history as an anti-inflammatory aid, and is now used concurrently with chemotherapy to combat its negative inflammatory effects. A widely known function of bromelain is its anti-tumoral abilities, linking the use of this enzyme to cancer therapy. Unlike chemotherapy, which has deleterious effects on all cell types, bromelain targets only disease cells based on their inflammatory nature. No lethal dosage of the enzyme could be determined in animal models, for which dosages significantly exceeded those given to humans in treatments. In this way, the non-toxic aspect of the enzyme is emphasized, providing justification for further research of this treatment that could be significantly less harmful to cancer patients. Bromelain alters the signaling pathway of tumor cells by removing the CD44 molecule from the surface of the cells. This signaling molecule is responsible for adhesion of cells, recruitment of lymphocytes to the inflammation site, and overall cancer growth and metastasis. Because of bromelain's ability to remove this molecule from the cell surface, aggregates of tumor cells, and therefore tumors, are not able to form. The Proteomics Laboratory at the Indian Institute of Toxicology Research has recently conducted 2 studies analyzing the effects of bromelain in the epidermoid carcinoma cells of mice and humans. Their findings detail a more specific pathway of these specific cancer cells for which bromelain decreases proliferation of signaling. In this way, the enzyme is not only preventing tumor aggregation, but inhibiting the expression of cancerous DNA. In both mouse and human models, the nuclear factor kappa B, abbreviated NF-kappa B, inflammatory pathway is integral to the proliferation of cancer cells. NF-kappa B is a heterodimer protein consisting of multiple subunits. In its inactivated form, the molecule is linked to an I-kappa B alpha protein and sequestered in the cytosol of the cancer cell. Activation of the complex occurs when the I-kappa B alpha protein is phosphorylated by a kinase, leading to degradation of the complex and subsequent release of the NF-kappa B. NF-kappa B is then translocated to the nucleus, where it binds to DNA and in doing so regulates gene expression through binding to promoter regions in the targeted gene. Transcription of the gene can therefore occur at a faster rate, subsequently increasing the rate of formation of mRNA for replication. Bromelain inhibits the translocation of NF-kappa B from the cytosol into the nucleus by preventing phosphorylation of I-kappa B alpha. Normally, I-kappa B alpha is phosphorylated at its serine 32 residue, but Western blotting of epidermoid carcinoma cells reveals bromelain's prevention of this phosphorylation while retaining the concentration of I-kappa B alpha, implying that it is not destroyed by bromelain, only inhibited. Therefore, NF-kappa B cannot be released, and the subsequent steps of the pathway are inhibited, preventing the gene expression necessary for the replication of cancerous cells. The ultimate effect of bromelain's activity is apoptosis, or programmed death of tumor cells. The result of the death of these cells are decreases in both size and number of tumors in mice with cancer given bromelain compared to cancerous mice that were not. As seen in the above diagram, increasing concentrations of bromelain correspond to increased apoptosis in both epidermoid carcinoma, abbreviated A431, and melanoma, abbreviated A375 cells. The discovery of this ability of bromelain to prevent the cancerous cell proliferation implies that it not only contains anti-tumor promoting, but also anti-tumor initiating abilities. In this way, bromelain increases its attractiveness as a candidate for further research in that it demonstrates multiple mechanisms through which it can be used to combat cancer cells. Further oncological research regarding bromelain should be directed towards the ability to use this enzyme as an independent anti-cancer medicine, rather than merely a cofactor of chemotherapy. Additionally, further study is needed in relation to the structure of bromelain and its mechanistic activities in pathways of other forms of cancer besides that of the epidermis. Published research lacks diversity in this respect, as many existing studies involve only cancer of the skin, which is relatively easy to study. However, now that the mechanistic pathway and effects on signaling proliferation are understood for these specific cells, clinical research has a well-defined direction for future study. This compound has been proven to be naturally occurring and non-toxic, and has demonstrated therapeutic potential in multiple facets of cancer growth and development. In this respect, bromelain can be deemed a worthy candidate for continued research in pursuit of providing cancer patients with a non-harmful and effective treatment.